So, one of the things I did this morning before class was that I made quite a few changes to the PowerPoint slides for the male reproductive system, which I believe is chapter 27, I think. Is that the right number? Anyway, I shortened it up quite a bit, and I try to simplify it so we could get through everything I wanted to today, so we didn't run out of time, because I do want to spend a full class period on the female reproductive system, because it's actually a lot more complicated than the male reproductive system. Okay, so if you would answer this question here. You count, let's see if everybody's answered. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. I think everybody must have already answered. So this is one that everyone should know. So we went over this in lab. And of course the correct answer is the testis. That's called the primary sex organ. That's the organ that produced the male haploid cells, which are the sperm cells. Those sperm cells, which have 23 chromosomes, combines with the secondary oocyte of the female, which also has 23 chromosomes. That returns the chromosomal number to 46, which is the normal number of chromosomes in all of our somatic cells. Okay? So, today we're going to take another look at the male reproductive system and also look at some of the physiology of the male sexual response. But first I want to go through again and review the process of spermatogenesis. You should be pretty familiar with this already because we went over this in lab. You also took a quiz over some of this. So I decided to use a different figure that shows spermatogenesis than the one that I used in lab. I didn't like the one I used in the lab. It was an artist illustration, and it just doesn't look like what it does in real life. So remember that the sperm cells are produced within the walls of hollow tubules that are located in the testis called seminiferous tubules. Excuse me, seminiferous tubules. These are microscopic tubules, and they're highly folded up in different sections of the testis. Each section is called a lobule. And the cells that start out the process up here, the cells that start out the process are diploid cells, which means they have 2 complete sets of chromosomes. Those are called spermatogonia. Those spermatogonia are located right under the surface. So here's some good examples of some of them right along here. Just underneath the basement membrane of the seminiferous tubules. And those cells are constantly multiplying by mitosis to produce more spermatogonia. And those cells that divide by mitosis are called type A spermatogonia. But some of the products of that cell division become type B spermatogonia. And it's hard to tell them apart because they look the same as type A spermatogonia. But some of those newly produced spermatogonia become type B spermatogonia, and then they start increasing in size and taking up nutrients. And when they start increasing in size, they become primary spermatocytes. Now, the conversion of a type B spermatogonium to a primary spermatocyte is not a result of cell division, not directly. The type B spermatogonia is simply increase in size, and they start moving closer to the lumen, or the hollow portion of the seminiferous tubule. And they're moved towards the lumen by the sustentacular cells, or the Sertoli cells. Okay, so it's actually the primary spermatocyte that then begins the process of meiosis, which is the type of cell division that produces the haploid sex cells, which in the case of the male is the sperm. So, the primary spermatocytes divide in the first division of meiosis to form two secondary spermatocytes. And in the process of meiosis 1, the chromosomes get segregated into two separate groups in the two secondary spermatocytes they're producing. So that reduces the number of chromosomes by half. So the secondary spermatocytes, the first stage that are haploid cells. Okay. Then each secondary spermatocyte divides again in meiosis 2, which is very similar to mitosis, almost identical to mitosis. The only difference is that in meiosis 2, you don't have a prophase stage. Rather, there is one, but the nuclear envelope doesn't have to break down in prophase 1 or prophase 2 of meiosis. In any event, each secondary spermatocyte then divides into two cells called spermatids. So you get two secondary spermatocytes produced in spermatogenesis, and you get a total of 4 spermatids. Those spermatids, though, at first don't have a tail, and they have a lot more cytoplasm than the sperm cells do. So before the sperm cells become mature, the spermatids have to go through a process called spermiogenesis. And again, this isn't a result of cell division. Spermiogenesis is just a result of what biologists call differentiation. As differentiation just refers to a change. They just change in structure. And so during the course of spermiogenesis, the spermatid grows a tail, which contains a flagellum, and it loses most of its cytoplasm. And the way it does that is actually the sustentacular cells, which these cells are attached to, actually pinch off a bag of cytoplasm. And so the mature sperm cells have very, very little cytoplasm. Most of it is just a thin layer around the head of the sperm, which contains the sperm nucleus. And then along the tip of the head, there's an acrosome, which contains lytic enzymes that allow the sperm to penetrate the outer layers of the secondary oocyte. So, at the end of spermiogenesis, you have 4 mature sperm cells, and each sperm has a head, which contains the sperm nucleus, a midpiece, which contains a spiral-shaped mitochondrion, and then a tail, which contains a flagellum. Okay. So going back to, this is an actual photomicrograph of a cross-section through a seminiferous tubule. These larger cells, like right here and here, those are primary spermatocytes, which form from the type B spermatogonia. When they divide, they form a smaller cell. There's one here, one here. Those are the secondary spermatocytes. And then they divide to form spermatids. And I don't know if you can see, I don't see too many spermatids in this particular one. Sometimes the development of these stages is sort of synchronized within a single seminiferous tubule. So you'll have some tubules that may have lots of spermatogonia and lots of primary spermatocytes, but very few secondary spermatocytes and very few spermatids. You might have others that have fewer primary spermatocytes, but they have lots of secondary spermatocytes and spermatids. So this one is kind of hard to see any of the spermatids, but if you looked at an another seminiferous tubule, you might see a lot of spermatids. All right. So, besides those different stages of development of the sperm cells, another important cell that's located in the walls of the seminiferous tubules are the sustentacular cells, which are also known as Sertoli cells. They have a number of important functions. One of them that I think I forgot to talk about in lab is that the sustentacular cells have kind of cytoplasmic extensions, sort of like if you think of my body as a sustentacular cell, so if I'd stick out my arms out, sustentacular cells sort of have arms that stick out, and they make contact with arms from adjacent sustentacular cells, and that forms a blood-testis barrier. So that prevents any potentially toxic substances that might be in the blood from coming in contact with the later stages of sperm cell development. So that isolates the spermatids and the mature sperm cells from substances in the bloodstream. So the sperm-testis barrier is sort of similar to the blood-brain barrier, which isolates some of the neurons in the brain from the blood, which could potentially contain toxic substances. Also, the developing sperm cells from the spermatogonia through the mature sperm become attached to these sustentacular cells by way of tight junctions, and then the sustentacular cells gradually moves them towards the lumen of the seminiferous tubule. So that transports them to the lumen. Once the sperm cells are mature, they're released from the sustentacular cell, and then they travel through the lumen, and then eventually they go out of the testis and into the epididymis. In addition, during spermiogenesis, the sustentacular cells also remove excess cytoplasm from the spermatid to form the mature sperm cell. And as we'll see today, the sustentacular cells produce a hormone that's involved in regulation. It's involved in the hormonal regulation of the reproductive system in males. And that hormone is called inhibin. And we'll take a look at what inhibin does here in just a little bit. Okay. So, any questions? Okay, and then finally there's one other cell that's located in between the seminiferous tubules. And these isolated cells in between the seminiferous tubules are called interstitial cells. They used to call them interstitial cells of Leydig, but when anatomists decided to do away with eponyms, which are names of structures named after people, they just dropped the Leydig part and they just called it interstitial cells. So these are important because they secrete testosterone. And without testosterone, spermatogenesis cannot occur. You have to have kind of a sea of testosterone in the seminiferous tubules in order for spermatogenesis to occur. Plus, testosterone also produces the male secondary sexual characteristics such as the deeper voice, a higher muscle mass, greater muscle mass, and also facial hair. So this is actually a photomicrograph that I took of one of our prepared slides. So you can see a spermatogonium right here. There's another one here that I'm pointing to. Those are the diploid cells that are constantly multiplying by mitosis. Some of the end products of mitosis become type B spermatogonia, and then they grow into a primary spermatocyte, which are these larger cells right here. Okay? Then the primary spermatocytes divide in meiosis 1 form secondary spermatocytes. So these lighter colored cells right here, those are secondary spermatocytes. Okay, now on this particular photomicrograph, you can't see any spermatids, but I think you can on this next one. Yeah, this is from a different seminiferous tubule. So these little dark specks up here, right here, down here, those are spermatids. Notice that these right here sort of looks like a larger sperm head, but they don't have tails. You can't see any tails attached to them. So those are spermatids that are just beginning the process of spermiogenesis. And then out here, these little thread-like things, those are the tails. This looks like a late spermatid right here, but it's kind of hard to tell because the picture's cut off. But eventually they grow a tail, they lose most of their cytoplasm, and then they become a mature sperm cell. So, in addition, right here you can see the nucleus of a sustentacular cell. You can usually tell that it's a nucleus of a sustentacular cell because you see a dark spot, which is called the nucleolus. That's where RNA is being synthesized. And then going back over here, there's a cell right here. That's an example of an interstitial cell. Interstitial cells are what produce the testosterone. Okay, then the next topic is the hormonal regulation of reproduction in the male. Now, hormonal regulation of reproduction in the male is a lot simpler than it is in the female, because once this begins at puberty, it just pretty much continues the same way for the rest of life. So males can produce viable sperm basically up until the point of death, because those spermatogonia are continually multiplying. So they never run out of spermatogonia. So this whole process starts out at an age of about 10 to 12 in boys at puberty. That's when puberty begins. And when puberty begins, the hypothalamus begins releasing gonadotropin-releasing hormone. And gonadotropin-releasing hormone travels from the hypothalamus down through the hypothalamo-hypophyseal portal vessel, or portal vein, down to the pituitary. And that causes the pituitary to release two hormones. It causes the release of follicle-stimulating hormone and also luteinizing hormone. Those are the same two pituitary hormones that help regulate reproduction in the female. But in the male, they produce slightly different effects. So in the male, luteinizing hormone stimulates the interstitial cells to produce testosterone. So you need to remember LH stimulates testosterone production. But testosterone has to be in the right place in the testis in order to stimulate spermatogenesis. And so to make sure that the testosterone gets into the seminiferous tubules, FSH stimulates the sustentacular cells to produce an androgen binding protein. And that's the androgen binding protein is produced in the seminiferous tubules, and it binds testosterone. And that holds the testosterone in the seminiferous tubules so that there's a high enough concentration of testosterone to stimulate spermatogenesis. So you need both LH and FSH to stimulate spermatogenesis. If if you have just LH alone, that'll stimulate testosterone production, but without the androgen binding protein that's produced by the sustentacular cells, the testosterone won't do any good. Okay? So in the presence of LH and androgen binding protein, testosterone stimulates spermatogenesis. Okay? Now, in addition, the sustentacular cells also produce the hormone inhibin. And inhibin has a negative feedback effect on the pituitary, and it mainly inhibits FSH. And so if spermatogenesis, if spermatogenesis is occurring too slowly, then the sustentacular cells will decrease the release of inhibin. That allow more FSH to be released, and the more androgen binding protein you have, the more testosterone will be bound within the seminiferous tubules. That will speed up spermatogenesis. If too many sperm cells are produced, then the sustentacular cells will increase inhibin release, and that will inhibit FSH release. That'll decrease androgen binding protein, and that will slow down spermatogenesis, so you don't have too many sperm. Okay. In addition, testosterone also has negative feedback effects on both the hypothalamus and the pituitary gland. And so that ensures that testosterone levels don't get too high. Because one disadvantage of testosterone is that it does tends to increase aggression in males, makes them more aggressive. So you don't want testosterone levels to get too high. So testosterone has a negative feedback effect, which primarily affects LH, whereas inhibin primarily affects FSH. FSH controls the number of sperm cells that are produced, whereas testosterone has a negative feedback effect, which regulates levels of testosterone independently of FSH. And besides having a negative feedback effect, testosterone also produces your male secondary sexual characteristics. It also, during adolescence, it stimulates the growth and development of the secondary sex organs like the penis, and it also stimulates libido, or sex drive. Okay, any questions? It might seem kind of complicated, but believe it or not, it's a lot less complicated than hormonal regulation of the menstrual cycle in women. Okay, next we're going to take a look at hormonal regulation, or I should say neural regulation of male sexual function. Okay. So sexual function during coitus or sexual intercourse in males involves 4 different phases. The first phase is excitement. That increases blood flow to the penis, which causes the penis to become erect. Also increases muscle tone and increases the heart rate, blood pressure, and the respiratory rate. After the initiation of excitement, there's a plateau phase in which heart rate, blood pressure, and respiratory rate continue to be elevated for a few seconds to a few minutes before orgasm. Then you have orgasm and ejaculation. It's a short reaction that results in expulsion of the semen. And then following ejaculation, you have resolution, in which blood flow to the penis is reduced and the penis becomes flaccid. So, at the beginning of sexual excitation, and this can be initiated either by tactile sensation of the genitalia or even thoughts. Just thoughts from higher brain center can also initiate excitement. And the excitement in plateau phase mainly result from parasympathetic stimulation. So the parasympathetic division of the autonomic nervous system releases acetylcholine onto the arteries that supply the penis with blood. And acetylcholine causes the blood vessels of the penis to release nitrous oxide, which is a gas. This nitric oxide actually acts like a neurotransmitter. And nitric oxide causes an increase in the concentration of cyclic GMP. And that cyclic GMP then causes vasodilation of the deep arteries of the penis, which supply the corpus spongiosum and the corpora cavernosa. And so the corpus spongiosum and the two corpora cavernosa become filled with blood. That increases hydrostatic pressure, which causes the penis to become erect. Now, there is a drug that some males use that have a hard time getting an erection. It's called Viagra. There's another one that's called sildenafil. And the way that Viagra works, and Viagra actually started out being tested as a drug for treating people that were having heart attacks or that were having angina pain. Angina is when blood flow to the heart is restricted. And because Viagra causes vasodilation, and so they were testing it to see if it could cause vasodilation of the coronary arteries in your heart, and which would increase blood flow back closer to normal. Well, when they were testing Viagra, which actually its name is sildenafil, Viagra I think is the brand name, some of the men reported that they got better erections. And so they decided, well, let's just use it for that then instead of the heart medication. So I think it's kind of interesting. The way that Viagra works is it inhibits the enzyme that breaks down cyclic GMP. And it's cyclic GMP that causes the vasodilation of the deep arteries of the penis during the excitement phase. And so by inhibiting the enzyme that breaks down cyclic GMP, that increases the concentration of cyclic GMP in the walls of the arteries that supply the penis, and that increases blood flow to the penis. And so that helps men that have a hard time getting an erection to get an erection. So the main thing to remember about the excitement and plateau phase is it's mainly a result of parasympathetic stimulation. And that can be affected by either tactile stimulation of the genitals, or it can even be caused by thoughts from the higher brain centers of the cerebrum. And then the next phase is orgasm and ejaculation. That's a short reaction that results in the expulsion of semen. The first step in ejaculation is actually called emission, spelled E-M-I-S-S-I-O-N. Emission refers to the movement of the sperm cells down through the ejaculatory duct and into the prostatic urethra. And that's a result mainly of sympathetic stimulation. So just before ejaculation, the sympathetic division starts stimulating the smooth muscles that are in the walls of the ductus deferens. And that causes the sperm to move down into the prostatic urethra. And as the sperm cells move down the prostatic urethra, then the prostate and the seminal vesicles add secretions to the sperm to form the mixture of sperm and fluids called semen. So that movement of semen then into the proximal portion of the urethra triggers a spinal reflex that causes spasmodic contractions of the muscle that surrounds the base of the penis, or the root of the penis. And those rhythmic contractions then expel the semen from the penis, which is called ejaculation. Okay. So this whole process of sexual function in the male requires the use of the parasympathetic division to initially cause erection. It also requires the action of the sympathetic division, which causes the semen to move down into the urethra, and then it also stimulates the release of secretions from the prostate and the seminal vesicles. And then that triggers a reflex which involves somatic motor fibers that stimulate skeletal muscles that surround the base of the penis. So there's three different kinds of nerves involved. There's both divisions of the autonomic nervous system, as well as motor neurons of the somatic division. Okay? So it's a fairly common, or it's a fairly complex process. And so it's no wonder that sometimes things go wrong and men have trouble with sexual function. All right. Any questions? Okay, then finally, the last stage is resolution. This is basically where the penis just goes back to normal, becomes more flaccid and soft. Resolution also results from sympathetic stimulation. That causes a constriction of the internal pudendal artery, which reduces blood flow to the penis. So once you cut off the blood flow to the penis, and the veins that drain the penis actually dilate, and then blood flows out of the penis, and then the penis becomes flaccid again. So that's referred to as detumescence. All right, so that's about all I have then on the male reproductive system, but I do have some review questions. Okay, so this first one has to do with male sexual function. And it says the expulsion of semen occurs when the bulbospongiosus muscle is stimulated by what kind of nerve? So what kind of nerve is involved in ejaculation? I'll give you a hint, okay? This bulbospongiosus muscle is a skeletal muscle that surrounds the bulb of the penis. Okay? That should give you a clue as to what kind of nerve that supplies it. So this is kind of the very last stage, right during the moment of ejaculation. Okay, let's take a look at everybody's answers. Okay, we've got a mixture of answers. So remember that erection results from parasympathetic stimulation. Okay, so it's not parasympathetic division. The sympathetic division causes the sperm cells to move out of the ductus deferens and down through the ejaculatory duct and into the prostatic urethra. That's called emission. And then ejaculation then is a result of a reflex that stimulates somatic motor neurons. So it's the somatic motor neurons that stimulate this skeletal muscle that cause ejaculation, which refers to the expulsion of the semen. All right, that was kind of a tough one. Okay, whoops, what is going on here? I don't know what happened. This looks like this question from an earlier chapter. It's not supposed to be in here. Here we go. I don't know what happened there. Okay, so this says blood is forced out of the penile lacunae. Those are the spaces inside the erectile tissues in the penis. By actually, I wouldn't worry about this one. You don't need to know this. I didn't go over this. So the answer is trabecular muscle. You don't need to go into it that much detail. So just focused on what I went through in here. Don't worry about this question. So, like I said, I went through and I simplified things quite a bit. Decided you didn't need to know as much detail.
One bonus point if you got that one right. No.
What's that?
One bonus point.
Oh,
if you got that right.
well, you can give yourself a bonus point, but yeah, I'm not going to do that. Okay, here's a better. This one is a relevant question. It says erection of the penis occurs when nitric oxide causes what artery to dilate? Most of what I skipped over was the diagrams that show the blood vessel, all the blood vessels that are involved, and the nerve, and where the nerves are located. As far as I'm concerned, it doesn't matter where they're located in the penis. It's just what type of nerve is involved in each of the stages. So most of you put deep penile. That makes sense. That's the only nerve that is located in the penis. So it is the deep penile artery. Okay. And then another one. This one has to do with spermatogenesis. The cells in the seminifer tubules that multiply by mitosis are what type of cell? These are one of the very, very first stages. ones that divide by, remember, mitosis is the more common type of cell division that occurs during normal growth and repair of our body. And the cells that do that are the very first ones. Those are the type A spermatogonia. So the ones that undergo meiosis are the primary spermatocytes and the secondary spermatocytes. So the primary spermatocytes undergo the first division of meiosis. The secondary spermatocytes undergo the second division of meiosis to become spermatids. Yes?
Are type B spermatogonia the ones that just grow? They don't.
Yeah, they grow and they become the primary spermatocytes. Yeah.
And then type A, to become primary, have to undergo mitosis.
The what?
If type A wants to become primary, they have to undergo mitosis.
Yeah, they have to divide, and some of those products of mitosis become type B spermatogonia. Yep, yep. Now, it's probably not even known what determines which of those end products of mitosis become type A and which ones become type B. There's probably some signal, but I don't know if anybody knows what that is yet. So. All right. Okay, I think this is probably one of the last ones. This is a good question. It says, which of the following cells are not haploid? In other words, which ones are diploid? There's actually two different stages that are diploid, and then the rest. There's two stages that are diploid, and the rest are haploid. Okay. So, yeah, the spermatogonia and the primary spermatocytes, they have not yet undergone meiosis, and so it's just the spermatogonia and primary spermatocytes that are diploid. The rest of them are all haploid. Okay. All right, I think that's the last one. Let's see. Yep, that's the last one. Okay, so Monday then we will have class. Don't forget we have class on Monday. And then we'll go over the female reproductive system.